Imagine being able to solve a crime with just the touch of a finger. In the world of forensic science, that's not just imagination, it's reality. Fingerprints, those tiny ridges on our fingertips, have become one of the most powerful tools in criminal investigations. But have you ever wondered why fingerprints are so reliable, and how they've come to play such a crucial role in forensics? Today, we're diving deep into the science of forensic fingerprinting, uncovering the secrets behind those unique patterns that make every one of us truly one of a kind. Let's start with the basics. What exactly is a fingerprint? A fingerprint is the impression or reproduction left on any material by the friction skin of the fingers. This friction skin is the specialized skin found on the inner hands and fingers, and on the bottoms of the feet and toes. It's characterized by alternating strips of raised ridges and furrows arranged in a variety of patterns. These patterns aren't just random, they're formed during the third or fourth month of fetal growth, around 120 days into gestation. As the skin develops, small islands surrounding pores begin to appear, which eventually form the ridges we see in our fingerprints. These ridges are permanent and unique, meaning that from the moment they form, they remain the same throughout a person's life, growing only as the body grows. Friction skin isn't just found on our fingers, it's also on our palms, feet, and toes. The impressions left by the friction skin on these areas are known as palm prints and footprints or toe prints. The structure of friction skin is fascinating. It's composed of various parts and layers that work together to create those unique patterns. The outermost layer is the epidermal layer, which includes the stratum corneum, or surface skin, and the friction ridges, furrows, and pores. This layer is about 1 to 2 millimeters thick. Beneath the epidermis is the stratum mucosum, also known as the Malpighian layer, which programs and forms the outer skin layer. Then we have the dermal layer, or the inner layer, which houses the dermal papillae, structures that determine the ridge pattern. The dermal layer also contains sweat glands, ducts, and nerves of touch, which are crucial for the sensory functions of our skin. One of the most important features of friction skin is its permanence. As we grow, our fingerprints grow with us, but their basic patterns do not change. Even as the skin deteriorates with age, the classification and identification of these patterns remain the same. However, friction skin is not indestructible. Injuries that penetrate the dermal layer, particularly through the dermal papillae, can permanently alter the ridges in the path of the injury. In contrast, injuries to the epidermal layer, such as paper cuts, will heal and the ridges will return to their original configuration. It's also interesting to note that self-induced injuries cannot remove all the ridges. Even if the pattern area alone were disfigured, classification might be affected, but identification or individualization would not. In fact, the disfigured pattern would likely become more unique, making identification easier. Skin grafts add another layer of complexity. In cases where a graft is applied, the old pattern might regenerate as the grafted skin wears away, or the graft area might remain smooth. However, a completely new pattern will not develop. Occupational wear can also wear down the ridges, but once the work stops, the ridges typically become distinct again. Even though friction skin is highly resilient, it can be temporarily or permanently altered by certain conditions. For example, in the latter stages of diseases like leprosy, the ridge patterns can be lost entirely. Allergic reactions can also cause temporary changes in the friction ridges, but once the reaction subsides, the ridges usually return to their normal configuration. This resilience is part of what makes fingerprints such a reliable tool in forensic science. Despite the potential for temporary changes, the core structure of the friction ridges remains consistent throughout a person's life. Now, let's talk about why fingerprints are so powerful in forensics. Every finger contains ridge detail that is unique to that finger and no other. These unique ridge details do not change, except in size, from approximately 120 days after conception until decomposition after death. This means that a set of fingerprints can be classified, filed, and searched with incredible accuracy, whether the system is manual or automated. Fingerprint identification is based on the premise that no two people, not even identical twins, have the same ridge patterns. This uniqueness makes fingerprints an indispensable tool for identifying individuals, whether it's for solving a crime, verifying a person's identity, or even studying genetic patterns in anthropology. From the moment they begin to form in the womb, our fingerprints are a testament to the incredible complexity and resilience of the human body. They are not just lines on our skin, they are a permanent record of our identity, with each ridge and furrow telling a unique story. In forensic science, fingerprints provide a reliable and powerful method for identifying individuals, helping to solve crimes and ensure justice is served. Whether you're fascinated by the science behind them or the role they play in criminal investigations, there's no denying that fingerprints are one of the most intriguing aspects of human biology.